Good morning. Thank you very much for the nice introduction, Dr. Walker. It's an honor and a pleasure to be here. Thank you. I would also like to thank the organizers. And it is my task to give an overview of the milk microbiome and neonatal colonization and the current state of knowledge as I see it. What I will be talking about, since I'm a physician, I will begin with health, breastfeeding and health, infant gut microbiota, breastfeeding and health, and those are topics we have already covered, but I can't help but mention those as an introduction to the human milk microbiome, what we know about its composition and origin. We will have separate talks about those, but I will touch upon that. And the last part of my talk is what we know about the significance of the human milk microbiome to infant gut colonization. And I can reveal at this point that we actually know precious little. And what we know about the clinical significance of the milk microbiome, we know even less, and perhaps some ideas for future research. There's only 4 buttons. Okay. Well, I know I'm preaching to the choir here, telling you that breastfeeding is probably one of the most important interventions to improve infant health. There was a beautiful paper in The Lancet a couple of years ago, which I recommend to everybody who hasn't seen it. Dr. Verhasselt alluded to this already yesterday. And based on the paper and based on the analysis in the paper, it's been estimated that the risk of death of an exclusively breastfed infant is only 12% of that of a formula-fed infant in the developing world. It's been estimated that breastfeeding could prevent about half of all diarrheal disease and about 1/3 of all respiratory infections. And consequently, exclusive breastfeeding, could save more than 800,000 lives annually. So it's really the perfect intervention we have at our disposal. And these results are actually kind of easy to understand. We heard beautiful talks yesterday about the protective molecules in human milk. So protection from infections during breastfeeding is probably largely mediated by those protective factors. I think it's also safe to say that in many parts of the world, an infant who is exclusive breastfed is not exposed to contaminated water to the extent that the formula-fed infant might be. So that might be a simple mediating factor as well. However, what is perhaps more interesting and more baffling is the long-term health impact of breastfeeding. It's been estimated that the risk of overweight and obesity is about 1/5 smaller in infants who have been breastfed in later life. It's been estimated that breastfeeding could prevent about 1/3 of all cases of type 2 diabetes, which becomes manifest at the age we start to gain weight and become middle-aged, so much later than breastfeeding. There's no data on supporting the notion that breastfeeding might actually have an impact of other metabolic manifestations such as hypertension or dyslipidemia. It's interesting that breastfeeding has been linked to reduced risk of leukemia, reduced risk of type 1 diabetes. The jury is still out in terms of asthma and atopic dermatitis and allergies. We heard beautiful talks yesterday. There's some suggestion about breastfeeding having an association with the risk of autistic spectrum disease and inflammatory bowel disease. But these are very, very difficult things to study. The diseases become manifest much longer, so we need long follow-up times. And as we all know, it's both practically and ethically impossible to do randomized trials with breastfeeding. So we have to rely on observational data, which is confounded after confounding. In terms of obesity, we know that the most important risk factor for childhood obesity is having an obese mom. We know that obese moms tend to breastfeed less, and shorter periods of time, they tend to give birth by elective C-section. They pass on their microbiota, they pass on their dietary habits, so it's really difficult to tease out what the impact of breastfeeding might be in the whole context. We heard yesterday that breast milk contains live cells and immunomodulatory molecules that might program or modulate immune maturation in the infant. And that's a hot area of research, but it's very, very difficult to tease out what molecules are important and which molecules play a role in terms of any given disease. It's also important to note that breastfeeding is not a controlled substance in the sense that different moms have different type of breast milk. And we heard that breast milk is different at different stages of lactation. But we also know that different conditions, maternal characteristics, have an impact on the content of breast milk. We know that allergic moms have different immune molecule concentrations in their breast milk than non-allergic moms. So it's pretty much a mess. So that's why I'm concentrating on a simple thing, the microbiota, which is actually by no means simple. We know, and what I find really interesting, that the gut microbiome has been related to the development of a lot of chronic diseases later in life. There's a list that kind of resembles the diseases that have been linked to breastfeeding. There's atopic disease, obesity and its comorbidities, necrotizing enterocolitis, which is important for me as a neonatologist, but also chronic inflammatory disease like inflammatory bowel disease, type 1 diabetes, some neuropsychiatric disorders. Now, showing that the gut microbiota in a person with a disease or disorder is different from a gut microbiota in a person who doesn't have the disorder is a long way from showing any causal relationship between the two. And what I find more interesting is the studies showing that very early life gut microbiota is already different in individuals who later develop disorders and diseases like obesity, overweight, atopic disease. And we're getting more and more studies like that, and we need studies like this, and they need to be combined with more mechanistic reductionist animal and laboratory models. But anyway, there seems to be a connection with the gut microbiota and chronic disease, and there seems to be a connection with breastfeeding and chronic disease. So we have two connections. Maybe they are the same connection to some point. Well, as we all know, breastfeeding has an impact on the gut microbiota. There is a graph from our Swedish friends from a few years back showing the gut microbiota development in infants during the first 24 months of life on the very general phylum level, and we see these huge changes taking place in gut microbiota composition, especially during the first months of life, which coincide with breastfeeding, the introduction of solid foods, and withdrawal of breastfeeding. There was a beautiful study in Nature last year from the TEDdy study showing that actually exposure to breast milk is the most important determinant of gut microbiota composition in infancy. And interestingly, it's actually the cessation of breastfeeding that causes significant changes, more than the introduction of solid foods. So there's something in breast milk that actually has an impact on gut microbiota. There are many papers showing differences in formula-fed and breast milk-fed infants, and we know that the gut microbiota of breastfed infants seems to be dominated by bifidobacteria. This has been the conventional wisdom for a long time, and it still seems to hold true. Interestingly, one of the members of the audience has shown a couple of years back that actually the impact of breast milk on gut microbiota is dose-dependent, and we know a great deal about the factors that promote the growth of bifidobacteria and certain other bacteria in the infant gut. We will have talks on human milk oligosaccharides, but there are also protein substances in breast milk that promote the growth of bifidobacteria. So what is the role of human milk microbes in this context? As I warned you, we don't know a great deal, but we know something, and that's what we're going into. So the human milk microbiome, this is something we have been talking about for about a decade and a half. To this date, about 200 bacterial species have been detected in milk. And this is just from a study we published this year. This is about, I believe, 40 moms. And if we look at the composition of breast milk microbiota. In this study, we see the major phyla, actinobacteria, Bacteroidetes, Firmicutes, and Proteobacteria present, and on species level, there's a great deal of Staphylococcus, Streptococcus, and there's also a great deal of, as of yet, unidentified bacteria. If you look at different studies, the human milk microbiome composition seems to vary. We don't know if it's because of the stage of lactation or the geographical location. There are a number of things that have an impact on on the milk microbiome, but also we need to think what we are actually measuring when we're measuring the microbiome in milk. The microbes in our milk samples may be derived from the mammary epithelium. There's good data showing that even the non-lactating mammary epithelium harbors a microbiome, and people who are interested in breast cancer are actually interested in the role of the the mammary gland microbiome in the development of cancer. But when we take samples, we also get bacteria from the skin, and actually we might get bacteria that originate in the infant mouth, because the child sucks on the nipple and we collect the milk that comes from the nipple, so we might actually be looking at infant mouth microbiota. Interestingly, however, in addition to Streptococcus and Staphylococcus, we constantly find obligate anaerobes like Bifidobacteria, Bacteroides, Clostridia in milk, which in my mind is some indication that we're actually measuring a microbiome specific to the mammary gland and not maybe so much the areolar skin. So we know something about microbes, factors that might have an impact on the milk microbiome. This is a study by Maria Carmen Collado and co-workers from a few years back, and they looked at the milk microbiome and they found interesting things. First, if we look at, they collected samples from a large number of sites in the mother. They collected maternal fecal samples, gut mucosal microbiome samples, skin samples, oral samples, vaginal samples, and milk samples. And in a simple principle coordinate analysis, they showed that the milk microbiome was very distinct from maternal feces, maternal skin, or maternal mouth, and even the birth canal. So there's a distinct microbiome. And interestingly, they had some moms who had delivered by elective C-section, and those milk samples were clearly distinct from the vaginally delivered mom's milk samples, and they resembled perhaps the skin. And when they looked at on the family and genus levels, it's a little bit small text, but here we have on the family level, vaginally delivered mothers who delivered vaginally, mothers who delivered with C-section, and elective C-section. And the elective C-section milk microbiome was distinct on the family and genus level, whereas the vaginally delivered mothers who delivered vaginally or the mothers who delivered by C-section after the onset of labor, there was really no difference. So this is an indication that something might actually be going on during labor that has an impact on the milk microbiome, or that labor is needed for a normal milk microbiome, and we'll get back to that. The other interesting observation in this study was that obese moms seem to have a less diverse milk microbiome. So the maternal metabolic state seems to have an impact on the milk microbiome. We do not know what the significance of that is in the development of obesity in the child. Other things that might have an impact on the milk microbiome. This is a small study we did with Dr. Bode, who's here in the audience. We measured from a small number of mothers. We measured the milk microbiome just by qPCR, certain selected species, and we measured human milk oligosaccharide concentrations. And it looks like human milk oligosaccharide concentration correlates with the bacteria in milk. And interestingly, things like Bifidobacterium longum were positively correlated with LNT, which is an oligosaccharide we know that specific bacterium is capable of utilizing. So it's tempting to think that already the milk microbiome is shaped by HMOs. We have really no data to show what the direction of causality is, but it shows that this is a complex system, and already in milk we see these correlations with microbiota and oligosaccharides. We need to continue that line of investigation. Other things, when we think of things that might have an impact on maternal microbiota and the maternal milk microbiota, is of course antibiotics. We saw the studies that the mode of delivery has an impact on milk microbiome. We started thinking maybe it's not the mode of delivery, maybe it's actually the antibiotic prophylaxis often given in C-sections. So we did a study, this was about 40, 50 moms who had delivered vaginally with or without antibiotic prophylaxis for group B strep, who had delivered by C-section or elective C-section with or without antibiotics. At that time in our unit, there was no universal antibiotic prophylaxis for elective C-sections. Now there is, but this was at a time where we're able to actually tease out the impact of antibiotics and C-section. And here we see in a principal coordinate analysis that the light blue and red dots really cluster together, and they are the vaginal delivery ones with or without antibiotics. So we don't see a whole lot of effect of the antibiotics, but the C-section babies cluster differently. So it's not so much the antibiotics, it's more the mode of delivery. When we looked in more detail, we see that the intrapartum antibiotic does have some effect, but these are milk samples that were collected at 1 month of age. So we see only traces of the antibiotic effect. The richness and diversity in the antibiotic-exposed moms is a little bit higher. So that's, again, one of the things that might have an impact. And intrapartum antibiotics is one of my favorite subjects because we know that we need intrapartum antibiotic prophylaxis to prevent group E strep disease. There's no question about that, even though the clinical studies are not so great. But after the adoption of screening practices and prophylaxis, we have seen a massive decline in group E strep sepsis. What I'm more worried about is the prophylaxis for C-section and for other reasons. And if you look at the Cochrane review on the impact, on the efficacy of intrapartum antibiotic prophylaxis or prior to C-section, they said that it prevents wound infections. It prevents uterine infections in the moms. And there's a horrible sentence that says the impact on the child is unknown. And for somebody who's taking care of the child, I think that's a horrible sentence. So we are in the process of looking at what intrapartum antibiotic might do to the child. And we are looking at a larger cohort. I don't have that data to share yet, but we did a small study of babies with infantile colic and compared them to healthy, non-crying infants, and we could see that all of the prenatal and perinatal factors that might be related to colic, we saw that intrapartum antibiotics increased the risk of infantile colic by a factor of 5. And the only protective factor we saw was exclusive breastfeeding. Again, it's intrapartum antibiotics, it's exclusive breastfed. We know that colic is somehow probably related to the gut microbiome. So again, more questions than answers, but I think this is a very important area of study. Okay, moving on. So we know that there is a microbiome in milk, and we know that it is different, related to, or associated to certain maternal factors, obesity, mode of birth, antibiotics, HMOs. What about, where does it come from? And I know there's a separate talk about this, so I'm not going into detail, but we have some fascinating ideas. As I mentioned earlier, we know that we find obligate anaerobic bacteria such as Bifidobacterium, Bacteroides, Parabacteroides, and Clostridia in human milk. And those are usually associated with the gut. So it's a tempting idea that there might be an enteromammary pathway for microbes as well. We know from clinical studies where we have studies in which probiotic bacteria are administered to the mom to prevent disease in the child or to prevent or treat mastitis. Investigators in Sweden and in Spain have been able to find those specific probiotic bacteria in milk samples. It might be contamination from the skin, but I think it's tempting to speculate that they actually go through an enteromammary pathway, end up in the milk. We have done quite a few studies on pregnant and breastfeeding women using probiotics, Lactobacillus rhamnosus GG, certain bifidobacteria, and I have to say we have tried hard, but we have not found those in milk. I would have loved to find those in milk, but we can't. But other investigators who are probably better than us have found them in milk, and I think that is really fascinating. If we think of the enteromammary pathway, there are nice studies showing that in experimental animals, in the perinatal period, the gut becomes more permeable. And you might remember I showed you data that being born by elective C-section without the onset of labor has an impact on milk microbiota composition. I think it's intriguing to speculate maybe that happens in humans too, and if you don't go through labor, your gut doesn't open up and the enteromammary pathway might not be functioning. This is all speculation, but I think it's really interesting speculation. This is an old study from 12 years ago, and investigators in France looked at maternal fecal samples in a small number of moms, 4 moms, maternal fecal samples, blood samples, milk samples, and infant feces samples. And with the methods they had available at that time, they were able to find certain strains of bacteria here, Staph epidermidis and Streptococcus salivarius. They found them constantly in maternal feces, in maternal blood samples, in milk samples, and in the infant gut microbiome. Again, and B. longum. And again, this is no direct proof that that is the way the child acquires those bacteria, but I think it's interesting, and they were actually able to stain those bacteria in maternal cells in human milk and in lymphocytes in maternal, or in immune cells in maternal blood. So I think this is a really fascinating area of investigation. Okay, I'm not going to talk more about the origin of the milk microbes since we have a separate talk on that subject. So now let's move on to what I was actually supposed to be talking about, the impact of the milk microbiome on infant gut colonization. And I've been stalling because there isn't a whole lot to say, but there are some nice studies. Whether we can prove that the microbes in milk actually colonize the infant or not is a matter of, we need really sophisticated studies. Thus far, we have really only done studies where we collect samples from the mom, from the maternal gut, from milk, and from the infant mouth and gut, and see correlations. And this is pretty much where we are at. This is one of the first studies that this was conducted in Finland already more than 10 years ago. This is about 60 moms and maternal fecal samples, human milk samples, and infant fecal samples were collected, and the presence and abundance of bifidobacteria were looked at by qPCR at that time, and constantly different species or groups of bifidobacteria were detected in all the sample types. So this might be an indication, okay, we find the bug in the maternal gut and in milk and the infant gut. This might be an indication that something is going on, but when they actually looked in detail, there was a strong correlation between the maternal gut bifidobacteria and infant gut bacteria, but there was really no correlation with the milk, with the abundance and presence in milk. So something is going on, but whether the bacteria in milk are just an innocent bystander and not actually a colonizing inoculum, it's hard to say based on this study. Moving on, this is a beautiful study from about 5 years ago. Only 7 mothers, but multiple techniques, a nice sample collection. Again, samples from maternal gut, milk, and infant gut, and 16S sequencing, so more advanced methods, and the data on the phylum level and also on the genus level. And the investigators find the same bacteria present in maternal feces, in human milk, and in infant feces. Interesting, there are some genera like Pseudomonas and Rastonia that are only present in milk, and they are not detected in the infant or in the maternal gut. But what the investigators did, they also cultured, and they, especially Bifidobacterium breve, they found viable B. breve in the maternal gut, in milk, and in the infant gut. Okay, again, circumstantial evidence, something might be going on. The author of this paper is in the audience, so I'm not going to spend too much time. A beautiful study, more than 100 mother-infant pairs, and they looked at the contribution of milk microbiome and the breast skin microbiome, and they could show that in a dose-dependent manner, it seems that they actually, through the use of a source tracker technique, actually in infants who are more than 75% breastfed, about 15% of the infant gut microbes originate from the milk. And what they did is really interesting. At the age of about 6 weeks, the gut microbiome of an infant resembled more closely to his or her own mom's milk than other milk samples, which is an indication that maybe the milk is actually a source of bacteria to the child. Moving on, this is probably the latest study from this year. It's less than 20 kids, but nice sample collection. They collected infant feces samples and from the mom both fecal samples and milk samples and mouth microbiome samples from the child as well. And if we look in detail, the infant feces here red, it's kind of all over the place as it tends to be, whereas the maternal feces is much more clustered. And we see that the milk microbiome is clearly distinct from that of the maternal microbiota, gut microbiota, and also from the infant gut microbiota. It's actually a bit closer to the oral microbiota, both in the infant and the child. And by source tracker, they showed that on day 2 of life, so very, very early, only about 5% of the gut microbiota originates in milk as per source tracker analysis. And at 6 months of age, the contribution of the milk microbiota to the gut microbiota is really negligible. It's 0.3%. So maybe it contributes more to the oral microbiota, which is an understudied subject. We published a study on the oral microbiota a couple of years back, last year actually. And we looked at 35 moms and children. We looked at the oral microbiota in the child and the breast milk microbiota. And we actually saw that they were quite similar in taxonomic analysis. But of the 22 genera detected in the study, only 8 were shared by both milk and the infant mouth, and the microbes shared were again Streptococcus and Staphylococcus. And when we tried to see whether a child's mouth microbiome was more closely related to his or her own mom than other moms, we weren't able to do that. So it doesn't look like the milk microbiome even contributes greatly to infant mouth colonization. So what goes on very early in life? You might remember from the previous slide that we showed that it's more important in the first days of life. This is a study we published a few years ago about 15 mother-infant pairs, and these were all children born by elective C-section. So we wanted to have a uniform group, and we actually wanted to avoid contamination during delivery. And we looked at, for the purposes of this talk, we looked at the maternal fecal microbiome, which were clustered nicely in a group and had very little resemblance with the other sample types. We looked at meconium in light blue, and we looked at infant feces at about 1 week of age in dark blue, and we see a shift during the first week of life. If you look close, you might imagine that the light blue dots shift in this direction and perhaps resemble colostrum more closely. So maybe something is happening during the first week of life. What is also important is colostrum. The colostrum microbiome is nothing like the maternal gut microbiome. And if we believe in an enteromammary system, it seems to be a very selective system, and this we see time and time again. What about later in the neonatal period? This is unpublished data from Henni Hiltunen, who's a PhD student in Turku. We looked at about 30 preterm infants between the gestational ages of 25 and 35 weeks, so it's a very heterogeneous group of preterm infants, and we did serial samples of the infant Gut microbiome, we took samples at 3 days of age, green, which are really all over the place. At 7 days of age, it's a prune-colored dots, and they start to cluster a bit more nicely. And 28 days of age, at 1 month of age, and this is when all babies were receiving breast milk. All of them were receiving some breast milk at 3 days of age, but at 28 days of age, all but one were exclusively enterally fed. And we see that the 28-day fecal samples start to cluster. So the chaotic initial colonization kind of draws toward a cluster at 28 days of age. The mom's fecal samples collected right after delivery on day 1 or 2, the first stool the mom passed after delivery, they cluster nicely, but they don't really resemble the infant gut microbiome. And then breast milk collected at 1 month of age, so at the same time as these yellow samples in red, and it's pretty much all over the place. If anything, it resembles the very early meconium microbiome, but we see really no clear connection with the preterm infant microbiome. Now, these are all infants in the NICU, so it's a very unnatural situation. I think all but 1 or 2 of the infants had received antibiotics at some point. But what we can draw from that is, again, the milk microbiome is nothing like the maternal gut microbiome, and it bears little resemblance to the developing infant gut microbiome. We are doing source tracker analysis as we speak. I don't have that data quite yet. Okay, so we see that something is going on. There's definitely live bacteria in milk, which the baby ingests. They might have some impact on the mouth microbiome. They might have some impact on the gut microbiome. Depending on the study, it's negligible to 15%. And we don't really know why they are there. I've toyed with an idea close to what Dr. Verhasselt told us yesterday, that maybe the bacteria in milk are not there to colonize. Maybe they are there to tolerize in the context of the tolerogenic milieu of the milk. We don't know. This is all speculation. Somebody said yesterday that nothing happens for no apparent reason. I think the microbes in milk are there for a reason. I think we have reason to believe that there is an active system by which they are presented in milk, and they must play some biological role, but we don't really have any grasp of what that might be. Now, if we think of the clinical significance of the milk microbiome, that also remains an open question. I think one of the first indications that the microbes in milk might actually have some clinical relevance is a paper we published last year, where we looked at, this was about 20 mother-infant pairs, and we looked at, again, the gut microbiota in the mom during last trimester of pregnancy and 1 month after delivery. We looked at the infant gut microbiome at 1 month of age and 6 months of age, and we looked at the milk microbiome in colostrum, the first days of life, and 1 month of age. And just by first glance, we see that and this is on genus level, we see that the milk microbiome is nothing like the infant gut microbiome. It's nothing like the maternal gut microbiome, which the maternal gut microbiome seems to be kind of stable in the last trimester of pregnancy and after delivery. The infant gut microbiome undergoes some changes, I would say, from 1 month to 6 months, but the milk microbiome resembles them not at all. And again, we see a lot of streptococcus, we see a lot of staphylococcus, some change over the course of lactation. But what is really interesting, when we looked at the number of antibiotic resistance genes in these samples, we see that infants actually harbor a large abundance of antibiotic resistance genes. And these are term healthy infants who have never been exposed to antibiotics. They actually have more antibiotic resistance genes in their gut microbiota than their moms. And halfway is the milk microbiome. There's a large abundance of antibiotic resistance genes in the mom. And when we put them on, again, in a two-dimensional space, we see that the maternal fecal samples, the antibiotic resistance genes in the maternal fecal samples are pretty stable during the last trimester of pregnancy and 1 month after delivery. And we see that the antibiotic resistance genes in the infants are clearly distinct, and these are not antibiotic-exposed infants, and they really overlap with a profile in milk. So I know that it's not a nice thing to say that milk might be doing some harm, but we actually might be passing our children antibiotic resistance genes in milk. Again, non-antibiotic exposed infants get the antibiotic resistance genes, at least partially, from milk, so they're paying the price for the past antibiotic use in the moms. What is a relief, though, is when we looked at the impact of breastfeeding on the number of antibiotic resistance genes, we see that almost universally, the infants who were breastfed at 6 months had less antibiotic resistance genes in their feces. So first I was saying that, okay, they get it from milk, but if they are breastfed, they have less. How does that work? The explanation we have for that is that most of those antibiotic resistance genes are in proteobacteria. They are actually in enterobacteria. E. coli is the most important carrier of antibiotic resistance genes in the infant gut, and the number of those bacteria is lower in breastfed infants. The breastfed infants, again in this study, harbored more bifidobacteria, which were protective of the antibiotic resistance genes. And again, my favorite subject, intrapartum antibiotics, increased the amount of antibiotic resistance genes. So from a practical point of view, we cannot help the past antibiotic use of the moms, and we cannot help how many antibiotic resistance genes the mom carries, and she might be passing them to her infant partially through breast milk. But the two wise things we can do to reduce that is not to use antibiotics when they are not needed and promote breastfeeding. Okay. Luckily, I can turn this into promotion of breastfeeding, but it was a little bit alarming at first. Breastfeed, promote bifidobacteria, decrease the abundance of E. coli and other enterobacteria in the infant gut, and you reduce the amount of antibiotic resistance genes. Well, I said this is the first indication of clinical relevance. Whether this actually has clinical relevance, we don't know yet, but it is alarming. So a lot of things are going on in terms of the milk. Okay. So what have we discussed? What have I tried to convince you of today? Well, first of all, there is such a thing as a human milk microbiome, and it seems to be affected by a number of maternal factors. The maternal metabolic state, maternal overweight, mode of delivery, especially if a C-section is performed without the onset of labor. It looks like the mammary gland doesn't know if the baby is born by C-section or not, but the mammary gland seems to know whether the mother has experienced labor or not. Antibiotics seem to have an effect. What we don't know, in our preemie study, we had a wide range of premature infants, and we saw no impact of the duration of gestation between 25 and 35 weeks on the milk microbiome, but I think we need to study that more. The impact of gestational age, the impact of lactational stage, the impact of geographical area, those are all open areas of investigation. We'll have a talk about the origin of the microbes in milk, but I think there are good grounds to hypothesize that there is an active system by which maternal gut bacteria are transported into milk. Why that may be, we have little or no understanding of. How the milk microbes affect gut colonization is an open question. There's some indication they might be. There's some studies showing that they really have no impact. My pet hypothesis that they are at least partially there to tolerize the child to the microbiota the child is going to have. Absolutely no data to back that up. At this point, it's a hypothesis. And the whole clinical significance of the milk microbiome is an open question. And we can say that it's frustrating we know nothing about it. I think it's motivating that we know nothing about it, and we should really get to work and find out why the bacteria in milk are there. In the end, I'd like to thank all the people who actually did the work from our lab, which I showed. I borrowed a lot of stuff from a lot of authors, but these are the people who directly contributed to the papers from Turku. Thank you very much.
This is open for discussion, right?
Very Very interesting and very important studies. Thank you for sharing it with us. I have two important issues to consider. Really. One is that is the microbiome cell-free or cell-associated? Because there is such a large quantity of particularly cells, epithelial cells, macrophages, and others. Do you think what you see is really cell-free material, or have you made an attempt to see whether there's a lot of cell-associated organisms there? That's one question. And the second question I have is, there's a lot of antibody activity in the milk to many of the organs that you talked about. Have you looked at association of immune complexes and tried to break it down in terms of how much of organisms are antibody-free or antibody-associated in the milk?
Thank you. Those are excellent questions, and I think interrelated. We haven't done any systematic studies in that fashion. I think we need to do that. I think at this point, when people are relying on sequencing methods, they just isolate DNA, and we have ways of trying to see whether there are intact bacteria or bacterial DNA, but we should also be looking at whether the bacteria might be intracellular, as you say. And there was the old study from France showing that they actually can stain the bacteria in cells. And the next question would be that if they are free-floating, are they coated with antibodies? And I don't think, at least I am not aware of any study where that has been looked at, but that would have a huge impact on their effects and immunostimulation in the gut.
Jerry?
I'm actually continuing in the question which you made. We live with millions of different, trillions, quadrillions of different bacteria, but they are not free. All bacteria in our oral cavity in the gut are coated in vivo with antibodies, which is a factor which you never consider in any of the studies. And yet, as I showed yesterday, the interaction is mediated by specific antibody activity or carbohydrate interactions. Now, you showed us cells which apparently have bacteria in it. What cell type is it? Is it phagocytic cell? They express Fc alpha receptors. Those bacteria are swimming in oceans full of IgA. Have you ever considered that may have tremendous importance not only for internalization of bacteria in milk in phagocytic cells, but also in the child? Because bacteria coated with IgA certainly behave differently than a free bacterium in the gut. That would penetrate. Coated with IgA will not. So are any of you looking at the effect of antibodies in the colonization and protection, for heaven's sake?
Thank you. Your point is extremely well taken. I don't think anybody has systemically done that. We're in the very first stages, but that's a very important point we need to consider.
That will explain so many things.
Yes.
Thank you very much for your nice presentation. Today we are breathing a bit easily. Being as pediatrician, yesterday it was all the talks were, and in research level and molecular level, but today we're feeling a bit relaxed. And my first question is that you have told that giving mother to Lactobacillus reuteri and cerevisiae to the mother. So is there any difference if you rub this bacteria, probiotics, to the areola and the baby suck it directly, or if you give it to the mother and then coming all the traveling to the mammary cells? Is there any difference of the bacteria behavior if you give it directly to the areola or to the mother?
Well, this is really beyond my expertise, but I think if the mom ingested that, it's been processed by the mom's immune system, and it might be delivered in an immune cell, in a phagocytic cell, in an APC, and it might be coated with antibodies. And if you rub it on the areola, I'm not sure how well some of those probiotic bacteria survive, but I know there are studies improving child oral health where they have actually a pacifier, which releases the probiotic directly in the mouth. So I think the areola might be rubbing the areola, which I do not recommend, might be a similar approach.
I have a second question. As you have mentioned about premature babies, in many cases of premature babies, the mothers are sick. Maybe it's eclampsia, and or multiple pregnancies with multiple premature babies, and they are really, devoid of breast milk is not available. So sometimes we use a surrogate mother, but though the composition of the milk is not appropriate for the premature baby if it is collected from the surrogate mother. In that cases, milk fortifier can be used for premature babies. But I do not know the Nestle. This question may go to the Nestle too. They have preparations for premature formulas, but they are not coming up with the milk fortifier so that we can use the milk fortifier and the milk from the surrogate mother to provide for the premature babies whose mother is really sick or eclamptic. So Nestle can answer this question, or you?
Well, that's a number of excellent questions. In our small preemie studies, we looked at a number of maternal factors and their impact on the milk microbiome, and we see nothing, depending on the cause of prematurity and a number of things, mode of birth, we see absolutely nothing in the premature baby microbiota. I'm not sure if the premature baby needs the microbiome, but I'm not familiar with using surrogate moms.
Milks fortifiers. Fortifier is not the microbes. This is the micronutrients.
Right. Yeah. Right.
Yeah.
Well, there's a point I want to make about using a surrogate mom, and you said if the surrogate mom has not given birth to a premature baby, that might not be appropriate milk from a premature baby. There is a notion that premature baby and moms of premature babies have different milk in composition, not only in microbiota, but in other things like EGF, and that's an adaptation to serve the premature baby. I have a very hard time believing that. In order for that to be an adaptation, it would have had to offer a survival benefit. And before the last century, I don't think any less than 32-weeker survived in order to actually have that evolutionary drive. I don't think the premature babies survived. There can't be an adaptation in human milk. I think it's more likely that it's a sign of an immature mammary gland at that stage of pregnancy. So I think, in my opinion, it doesn't matter if you use a donor milk or a surrogate mom, whether the donor delivered a premature baby. I think we should pay attention whether we give colostrum or early milk. We don't have a lot of donated colostrum, but we should pay attention at which stage of lactation the milk sample has been taken. Now, in terms of fortification, there are fortifiers you can use in human milk, and it doesn't matter if it's a, I'm not sure if I understand your question, because you can use them on a
It's not available everywhere. Milk fortifier is not available in our country.
Okay.
But the premature formulas are available, Nestle's are.
Right, okay.
Yeah.
I didn't appreciate that, and I'm a simple neonatologist. I'm sorry I can't help you, but that is a problem.
Carlos?
Yeah, in continuation with the idea of the probiotic not appearing in breast milk, I wonder if anybody has done like the labeling of the probiotic with nitrogen 15, for example, which is a non-radioactive isotope, and then seeing enrichment in breast milk just to see if the protein of the probiotic, might not go beyond past the lymphatic system, but it could contribute in some way to protein or something in breast milk.
That's an excellent question, which I can answer, and I direct it to Dr. Rodriguez, who will give the next talk. Maybe you can address it.
Maybe better after the next talk. Yeah. Okay. Ferdinand?
I would just like to clarify the availability of human milk fortifiers. Human milk fortifiers are globally available from 4 companies at least. They are available in liquid form and in powdered form. If they are not available in your country, it's an effect of year-long fighting with the regulatory authorities in the respective countries which do not accept the fortifiers. It is also something which is very difficult in terms of the interactions with NGOs. Some NGOs in the countries, mainly in South Asia, they believe that this interferes with breastfeeding. To a certain extent they are right, because the fortifiers are difficult to handle. The mother has to pump the milk and then mix it and give it in a bottle. So this is not something where companies are not willing to distribute fortifiers to the countries. It's mainly the interaction with the regulatory authorities which do not allow to have them in your country.
At the same they are giving permission for the premature formulas.
The formula is a different thing. A formula is a different category of special food for premature infants. One is a supplement and one is a whole nutrition. So the authorities see it in a different way.
Are there any more questions? If not, Sam, thank you very much.
One question. One question.
One
question, please.
You know, that light is shining right in her eye.
Yeah.
I can't see that. Go ahead.
Thank you for this very beautiful synthesis. I had one comment and one question. The comment was regarding the IgA and microbiota. So there are papers showing that the IgA, of course, shape the microbiota, but also influence the metabolism of the bacteria. So it really goes far. And then a question we had yesterday talks about evolution of breast milk and how is it spread among species. Have you ideas about or knowledge about microbiota in breast milk of other species? Is that also found? And
I have to confess, I have absolutely no knowledge of that.
thank you.
One question.
Are there any other questions?
Yeah.
Okay, go ahead.
In case of necrotizing enterocolitis, actually we don't give the best feed for certain period of time. On that time, could be probiotics be given for the prevention of the necrotizing enterocolitis?
Now that's a discussion no neonatologist wants to enter. Yes. Well, I believe there are very few instances when a premature baby shouldn't receive breast milk. And those are instances when we are afraid to use the gut at all. And I think at those times, I wouldn't give probiotics either. When the baby receives breast milk, I think if you are among those who believe in probiotics, whenever the baby is receiving breast milk, you can give probiotics as well. But if the baby is NPO for a reason related to the intestine, I wouldn't give probiotics. But that's my opinion. I'm not even sure if that's an expert opinion, but that's my opinion. I'm not sure if Dr. New has other insight.
I'll talk about it later.
Thank you very much, Sam.
Thank you.